Okay. All right, I think you can get started. Thank you again, everyone, for coming, and for those online who are watching. My name is Andreas Bonner. I'm Director of Informatics and Computational Biology at the Englander Institute for Precision Medicine. And today we're going to talk about precision medicine knowledge base, which is one of the components of the precision medicine, or as we call it, PMKB. First, what is precision medicine? So there are many definitions. What I like is the approach of medicine that delivers the right treatment for the right patient at the right time. And for doing that, this is inspirational to get information about many components, many data points, let's say, for each patient, including environmental, but mostly precision medicine started when we had a kind of a genomic, or let's say omics revolution a few years ago, where we could, technologies improved such that we could look into patients' genomes, and in particular, in this case, we're talking about cancer genomes, to see what kind of alterations, what kind of defects, let's say, these tumor cells have. And so the types of alteration that we are looking at are different types, as I indicated here. We have single nucleotide variants, so some typos in the genomes, in the tumor genomes, compared to what it should be, that alter the function. And also small deletions, so missing a few letters or adding a few letters. Copy number alterations, which is basically the same concept in a sense of indels, but on a much larger scale. And you can have deletions, so missing parts of the genome, amplification, where some parts are duplicated many times, one or more. Inversions, so reorganization, let's say, of the genome. And these also might give rise to what we call rearrangements or gene fusions, where you have portion of the genome that kind of move around in places they're not supposed to be. Typically, these alterations are detected. There are many different ways to detect those alterations, but we are talking in this case using these genomic pipelines that analyze sequencing data. And here's kind of a very brief schematic of what a genomic pipeline does. It starts from, there is a machine, let's say, after you extract your DNA from your sample, a machine will translate this biological samples into a series of letters. Some computational analysis would assign those letters to a particular patient and to a particular region in the genome. And eventually, at the end of the process, there is what we call the variant calling, or identifying basically those defects that I mentioned in the previous slides, either single nucleotide variants or the bigger copy numbers alterations. However, these pipelines now are being used more and more in the clinical settings. Researchers, let's say, are analyzing those data, and they come up with a series, or they identified by doing research studies what are the most important alterations that we want to see in the genome. Now we're moving towards, let's say, the clinical settings, where we want to sequence patients' tumors, for example, and identify those mutations, and then report them back to clinicians. The challenge here is an example of our reports for one of the whole exome, the sequencing tests that we do, is based on a whole exome sequencing, is to provide this information in a, let's say, manageable and understandable format. And here's where PMKB come to play. So at Weill Cornell currently, we have a few tests that are clinically run and approved by New York State. Some of them, like the first three here in this table, are what we call targeted panels that look at very specific areas of the genomes for specific disease types. AmpliSeq, for example, for solid tumor, it looks only at 50 genes. Myeloid is specific for hematological malignancies, the myeloid in particular, 40 genes, so very small. OncoSeq is a broader targeted panel still, but looks at solid tumors. And also, in addition, looks for a selected set of fusions from RNA, and it's kind of unique. And then we have ExactOne, which is one of the few, probably one of the two clinically approved tests using whole exome sequencing in the country. And it looks at 22,000 genes, so the entire, say, human genome, or coding part of the human genome. And in particular for patients with advanced cancer. These tests are fully integrated into the clinical pipeline. Once the results are available for particular patients, the molecular pathologists are reviewing those data, looking at these variants. Those variants are classified by importance. And in particular, in our case, we use 3 levels of importance, tier 1, tier 2, tier 3, and we're going to explain a little bit later. But the important part is that these association is done at the level of the analysis pipeline, and then associated to that classification, there is an interpretation that allows the receiving, let's say, oncologist treated a patient to understand the impact of each variance for that particular patient and creates these reports. So these interpretations, these are the key elements that are used for clinical activities. And the precision medicine knowledge base is basically our repositories of interpretations. And the rationale for us for building this is we want to have kind of a standardization of all these interpretations regardless of the specific genomic test that we ran out of the 4. So all of them are using the current PMKB or precision medicine knowledge base for delivery, basically those report back to the clinicians. So I'll stop here and I'll let Dr. Nam to continue and presenting how we basically generate the content of this in this PMKB.
Thanks everyone for coming. So typically what happens in the clinical setting is, we get the results back from the sequencers and we get variant calling. And we have this amazing database which links that variant to that tumor type, to that site of origin with interpretations and also tiering. So in cases in which we don't have that interpretation, we have to create our own, and that's kind of where you guys would also fit in. So this is the database, or this is the website where all the data is stored. This is the updated version that will be coming out soon, but we have a working version right now that's very similar to this. So you can see all the genes that are annotated, the variants of those genes, the interpretations for these variants, the tumor types, and the primary sites. Okay. So in the typical entry of a PMKB, you have the gene, BRAF in this case. You have the variants, so the hotspot is V600. So all those variants associated with disease oncogenic variants, the primary site, and the tumor type. So BRAF is oncogene, and these variants are seen in multiple types of tumors. In this case, it's showing tumor type of hairy cell leukemia, but it's also commonly found in other tumor types like melanoma or lung cancer. So the interpretations are gonna be dependent on what type of tumor it is and what the primary sites are. So in terms of the components of the interpretation, however, there will be sort of commonalities between these interpretations depending on the gene and the variant. So for example, the first sentence is gonna be what that gene function or the protein of that gene is doing in a biological setting. So BRAF is a member of the RAF family, and it plays an important role in the signaling pathway. And you can imagine that an activating mutation in this gene will be tumorigenic. So that will be often the case, the first sentence, and it will be common throughout tumor type and primary site. And then you go into the prevalence of that, prevalence of mutations in that gene for that particular tumor type. Here it's giving you information for hairy cell leukemia, but also some of the other hematopoietic malignancies. And you would only have to really focus on the tumor type and the primary site. For example, mutations of BRAF are found in X% of lung adenocarcinomas, and you wouldn't necessarily have to go into all the other solid tumors or anything like that. But in this case, they give percentages for the other tumor types, and it's kind of basically a hint at how important that gene is for this tumor type. So, for example, BRAF is described in up to 100% of hairy cell leukemia, so clearly a very disease-defining gene and variant for this tumor type, and also important for other tumors like Langerhans, histiocytosis, and so forth. And then the next part would be whether this particular variant is oncogenic or not. So V600 is an easy one where everyone recognizes as a hotspot, but so it's been very well characterized as a oncogenic variant. So that would be sort of a key statement that would, I guess we didn't really go into tiers yet, but that would distinguish whether it's oncogenic or not. And then the last sentence would be describing the role of therapy for this specific target and this specific tumor type. So there's a specific inhibitor for hairy cell leukemia, but this drug may not be FDA approved for other tumor types like lung cancer, even though BRAF V600E may be found in lung adenocarcinomas. So that's why it has to be catered to the specific tumor type and primary sites. I don't know if do we have another specific section where we talk about the tiers, or? Oh, you'll talk about it. So those are kind of the main components, I think. Yeah, so it's basically outlined here with that as an example. So the first sentence is going to be a brief statement of that molecular function of the gene or the product thereof, the role in its specific tumor types, and that's really mostly looking at the prevalence of mutations in that gene, and then how that specific variant promotes the tumor, or whether it does or not. And then you can go into the prevalence of that gene variant in that specific cancer type, and then whether that variant has a therapeutic implication for that tumor. So here is that interpretation kind of broken down into those parts, and so you'll see in the database, which is publicly available, that a lot of the interpretations follow that guideline, and sort of maybe not exactly in that order, but hopefully most of them will have all of these components. Okay. And then in terms of where to look for the information for these different parts of the interpretation, oftentimes that molecular function statement will be found in another interpretation, even within PMKB. that there are some other sources that are listed here that are also databases with interpretations for variants for various tumor types. So you could also access these other ones, but I would encourage you guys to go to the primary source. So like all of these are like PMKBs in which, you know, a human being sat down, wrote down interpretations based on a primary source. So always check back to the primary source, even if you gather information from OncoKB or JAX CKB and so forth. In terms of whether that variant is tumorigenic or if it's unknown, OncoKB and JAX, if it's not in PMKB, OncoKB and JAX are also good resources. But oftentimes they won't have information for that variant. In that case, you would have to go to the primary literature source, and that's in PubMed or Google, you know, pick up literature also. For prevalence, I find CBioPortal to be the most convenient database to look. I think it would be good if in your tutorial we have a session on how to get information from CBioPortal, but you could, is it in there? Yeah. Yeah. But basically, CBioPortal is a database in which these huge databases of sequenced tumors are imported into this database, including our data also. So you can see the prevalence of mutations in the specific tumor type that you're interested in to get the percentage of cases that have mutations in the gene that you're interested in. And then for therapeutic prognostic impact, you can again look at OncoKB, FDA-approved drugs, you know, clinicaltrials.gov, and Google again. So this is the OncoKB, which is across the street MSK's database. So it, you know, they've been sequencing for a long time now, so they have a pretty decent database. But again, I would encourage you to always look to the primary source. So this is the CBioPortal that I mentioned with kind of databases from these pan-cancer genomics initiatives. So they have you know, thousands of samples that are sequenced that you can tap into and see how many times a certain gene was seen to be mutated in a certain cancer type. Okay, and I'm sure you guys into PubMed. These are some of the, so, you know, just because something is published doesn't mean it's good data. So we like to cite reliable sources. So these are some of the more reputable journals that you should probably stick to. Although there are, you know, variants that are so rare that will be only reported in smaller journals, and in that case, that's what you'll have to use.
Thanks, Anna. Hi, everyone. I'm Monik. I'm the one that you've been communicating with. Really happy to see so many of you here today and see even more of you all zoomed in. So I'm just going to talk about some of the details of the PMKB crowdsourced pilot project. And from there, afterwards, you guys can ask me any questions that you might have. So start out with, what is the project? It's pretty simple. So this project is about taking driven and curious individuals, just like all of you, and getting you to help us author uniformly structured and up-to-date interpretations for gene variants in PMKB. Sound simple enough? Cool. So the question you might be having is, why? Now, as Anna has alluded to, many of the variants that we already have interpretations for on PMKB vary in terms of, quality, in terms of structure, in terms of relevance. Many of them may be out of date, sort of like that flip phone. And so we're really looking to enlist as many of you to help us harmonize the structure and update a lot of the interpretations with the most recent literature. So I'm just going to reiterate again, what are the goals of this study? On your end, from contributors, we would love for you guys to help us update the current non-generic interpretations that we host on PMKB to reflect the most recent literature, while also harmonizing the structure to a uniform structure that we will describe a little bit more in the end of my slides. On our end, us being here at the IPM, we're going to be assessing your performance. We want to understand, how do you all perform as non-clinicians, as non-experts, as non-professionals in terms of crafting clinical grade interpretations for these gene variants? And also we want to see, does crowdsourcing even work in terms of curating these sorts of clinical databases? And that's what we're trying to understand. So what's the structure of this study? So as of today, you can register to become a contributor to this PMKB project. You can, well, what I'll do is I'll send out an email and you all can go to the PMKB website. And if you have a Gmail, or if you want to link your current email with a Google account, you can register. And from there, starting on Monday, we will approve your access and you'll begin being able to start actually editing interpretations on PMKB. Once you are finished with an interpretation that you've written, you can submit it, and that will be sent to our team of molecular pathologists associated with this study, and they will review what you've written. What they'll do is they'll suggest edits, they'll give revisions, and they'll send them back to you. And then you all will incorporate those edits and revisions into your interpretations and then send them back to us. And this will keep going until it's clinical grade, when it's finally approved. And you will all receive points for submissions and for approvals, which will be counted towards the incentives. And I'll talk more about the points and the incentives in a little bit. So what's the overall timeline of this study? As I said today, you can start registering to continue registering over the weekend. We'll start opening up your all to have access starting on Monday. Even if you're not able to register over the weekend, even if you're not, approved for access by Monday, you can still register and still get approved later on. You all will be continuing to draft interpretations from next Monday until about June 17th. At that point, we'll ask you to stop submitting new variant interpretations, and we'll focus the last month of the study on, the revisions, the edits, and approvals. And the tentative date for the end of the project as of right now is July 14th. But it should generally be mid-July. Maybe a little later, maybe a little sooner, depends on how things go. So I'll talk a little bit more about interpretation drafting. To start, we want you guys to focus on non-generic interpretations hosted on PMKB. What is a non-generic interpretation, you may ask? Well, I'll answer that with another question. What is a generic interpretation? Generic interpretations that we have on PMKB are ones that say all the same thing. They all say, this gene is a known cancer gene. We don't want you guys to write any interpretations for those. We only want the non-generic ones, because those are the ones that are actually used by clinicians on a daily basis very often. After you've crafted your submission, which will be in the structure that I'm going to tell you about, you will submit them with the most recent and relevant references that you were able to find on PubMed or Google, or that you've pulled from these other databases. I will tell you, I will be posting a video tutorial on YouTube with this talk as well, which will walk you through the steps of crafting an interpretation. It'll be step-by-step, it'll be methodical, it'll tell you how exactly you can contribute. I should also say that for many of these gene variants, the jury's still out on whether or not they may be actionable. So there may be a gene variant where two recent papers may say it has clinical relevance, and another one that says it doesn't have clinical relevance. That's completely okay. If you're writing an interpretation for a variant that is like that, just include both references and submit them on PMKB. So again, this is just what a generic interpretation looks like. As I said, it says this gene is a known cancer gene. Don't write interpretations for these yet. If the project goes successfully, if I see you guys are all knocking all the non-generic interpretations out of the park, in a month, 2 months, we'll open these up for you all to start contributing interpretations for the generics, and you all can start just accumulating more and more points. So what is the structure of the interpretations that we would like you guys to submit? 4 points, just like what Anna said. In fact, these are the exact same 4 points that Anna had mentioned, just a slightly different order. Here we have, as the first 2 points that we want you to focus on, are the prevalence of the gene variants and their prognostic or therapeutic significance. Why is that? Well, that's because those are the most relevant bits of information for clinicians. That is what is actionable. That is what an oncologist is going to look at when they are reading an interpretation to try to judge whether or not they can tailor therapy based off of a tumor's mutational profile. And then afterwards, we can get into more of the detail of the molecular function of the gene. So this is an example of what an ideal interpretation will look like in that structure. As you can see, we got a one-liner here at the top, that clinicians can use. Short and sweet, and it's got all the necessary juice that they need. And down here, we got more information for people who want to learn more. So submitting. Once you are done drafting your interpretation, you've accumulated all the references that you think are relevant, you can submit it on the PMKB website. I'll show you how to do that in the video tutorial. Afterwards, we're going to ask that you fill out a brief, very brief Google form. And all you'll have to do is put in the link to that interpretation that you wrote on the PMKB website and log, is this a new interpretation I've written, or is it a revision to one that I've already written? Now I'll tell you, completing the Google form is required to receive points. Why is that? Well, we're going to be using it to track all of your submissions and to track pathologist comments on everything that you submit. This is what it looks like. As I said, short and sweet, shouldn't be too much of a hassle. So the review process. As I said, after you submit something, it will go to our team of molecular pathologists. They'll read what you've written, they'll look through your references, and they'll send back some comments saying, oh, hey, you spelt BRAF wrong. Or, you might want to include some more information regarding prevalence, or maybe grammatical edits. That will get sent back to you, and then it is incumbent upon you to edit your interpretation to reflect their comments. And then when you are done with your revision, you will, again, submit it on PMKB, and then resubmit the same Google form with the link to the edited interpretation. This is what it looks like on the other end of the Google form that you guys are going to be submitting. So as you can see here, these are the links to the interpretations. Those aren't real links, I made those up. And logs whether or not it's a new interpretation or a revision. And then here, start having pathologist comments and the date that they read your interpretation and made their comment. This will be publicly available, publicly viewable by all contributors. And that's on purpose. We want you all to be able to see comments on everybody else's interpretations. We want you to not only learn from the mistakes that you make and the iterative comments that you make or that you receive, but also on everybody else's. If you can identify patterns of mistakes that people are making, you can incorporate that into your own interpretations. You can learn from it, and you can make high quality interpretations that have a higher likelihood of being approved much more quickly. Make sense? Sounds good. Okay. After the approval process, or throughout the whole process, you guys will be getting points. What are these points? What are they good for? We'll break it down. So points are awarded based on the tier of the gene variant submitted. As Anna and Andrea have already mentioned, there are 3 tiers that we have for variants in PMKB. There's tier 1, tier 2, tier 3. And tier 1 is basically the most actionable gene variants that we have hosted. If a targeted therapy exists for that gene variant, it's tier 1, and so on and so forth. And you can see the nitty-gritty details on the PMKB website of what qualifies as tier 1 or tier 2. but they are all already assigned to a tier, all the variants that are hosted. Because we want you guys to focus on the most clinically actionable variants that we host, we are giving the most points for tier 1 variants. We want you guys to finish those first. After you submit, you'll get the points. Then, after you are approved for that submission, you will get an additional 10 points. Why is that? Well, we are trying to incentivize high-quality interpretations. We want you guys to focus your time on submitting a quality submission and not just on quantity. So, do some basic math. Let's say you spend 4 hours writing one tier 1 variant, interpretation. You'll get 4 points. It's really high quality. It might get approved right off the bat. You get an additional 10 points. That's 14 points. Let's say for another person in that time, they write 3 tier 1 variants. They get 12 points, but then they go back and forth for maybe about a week with pathologists iteratively improving until they finally get, an approval for that interpretation. That's 12 points versus 14 points. The 14 points were acquired much more quickly. Again, we want you guys to focus on writing high-quality interpretations right off the bat. I'll also say we're going to be reaching out to contributors every 2 weeks. So every 2 weeks, we're going to tell you what is your point system, what do your overall points look like, and how does it look relative to everybody else. And at that time, we'll also say, this is how many submissions were made to PMKB over the last 2 weeks. This is how the project is looking, and we'll ask for feedback. We'll also send an additional survey, again, very brief, very small, that'll be asking for some additional information so we can try to chart your performance and see how the study is going. And again, all these points that you guys are getting will count towards earning incentives. So what are the incentives? Well, of course, the most important incentives are to learn how to better interpret scientific literature. I think that that's relevant for all of you young, budding physicians and scientists. And even more importantly, every contribution that you make will make an immediate impact on patient care, both here at Weill and at hospitals across the world that use PMKB. If that isn't enough for you guys, we are also offering authorship. As of right now, we're offering authorship to people who are going to be in the top 50% overall points, of contributors. That's tentative. The sword swings both ways. If we don't get much contribution, then this study won't get published at all, and no one gets authorship. Not myself, not Andrea, not Anna, and unfortunately none of you. However, if this project is wildly successful, then we have no issue widening the scale to the point where 75%, 80%, even 90% of contributors who've made a meaningful impact in this project can be included as authors. Otherwise, we are also offering cash prizes, probably in the form of Amazon gift cards, to the top 3 contributors. I think as of right now, we've said $200 for first prize, $150 for second overall, and then third prize will be $100. If you want, you can donate that money to a charity of your choice. The IPM is partnered with a great one that's doing really good work. It's the Monaco Paul Beer Fund. We have some beneficiaries of that beer fund in the back row. And they would really love to see you guys, open your hearts and donate to charity. But if that's not what you want to do, it's not an issue. Go ahead and be selfish. So that concludes my presentation. If anyone has any questions, happy to take them. Can we also open up the Zoom for anyone who has questions via Zoom? How do I do that? Okay.
It's done.
Question? Okay. Can you read? Can you think critically? Can you do some degree of pattern matching? Okay, so that's probably enough to get started. And then, assuming you also know how to learn iteratively from comments and from seeing how other people have made mistakes and are improving from the comments that they receive, should be okay. But if you want to talk more about it, we can talk, offline. Anybody else? Questions? Yes. Yes.
So we just ran whatever tier we want to work on, and that essentially is powered for.
I'm sorry, I don't think I fully understand.
I'm sorry. So there's 3 tiers, so 1, 2, 2. Right? Tier 3 is the one with the 4 points. It's the, I guess, the hardest one that we have.
Tier 1 is the one with 4 points.
That
Yes.
we have to annotate. So once we select that, no one else can touch that until it's fully approved by the clinician and all of that.
So you're not selecting a tier, you're selecting a variant which corresponds to a tier. We just want you to focus on tier 1 variants as of right now.
So it's not assigned to us,
you just pick it. But the one that you pick, once you submit an interpretation for it, it's locked to you. That way, nobody else can come in and try to revise something that you've already written and try to steal your points. We've made it such that, you know, people who claim variants will be able to keep them. That being said, you know, if anyone is found to be engaging in nefarious behavior, like going into a bunch of tier 1 interpretations and just adding a space and saying, okay, I submitted a revision and, you know, these are all mine and I'm going to get all the points, you know, we're monitoring that and you'll be booted from the competition and won't be eligible for any of the incentives. Yes? To some degree. Well, once you submit, it's yours.
Oh, okay.
Yes.
Right. So then you have the thing we're worried about is, God forbid, I'm doing something in a sheet, and then we're both competing, and then 5 seconds, one of us clicks and submits, and it's like the other person kind of like screwed over. Yeah, because you're like writing for so long. Or like...
Fair point.
Because in the past project I did at the Mason lab, we got an Excel sheet and we got 10 different things and we had to annotate. Those were our specific kind and no one else can touch that and submit it. So maybe like there's a way that we can like name it before someone else does or something.
Yeah, I mean, that's fine. Does that answer your questions? Does anyone else have any other questions?
Correct me if I'm wrong. I guess the moment you see a variant and sorry, let's go. So if you see a variant and you want to edit that, you can immediately edit it. I think it's an edit interpretation if I remember correctly. The moment you have it, it's like you claimed it. Okay. So nobody else can use it or wait until you write almost everything and then just say, oh, this is mine, I'll submit, right?
Yes.
Okay, so the moment you basically say edit interpretation, of course.
Yeah, any other questions? Are there questions coming in on Zoom? Or
It was the same question. It said, how, once a variant is worked on, can others pick the same? And how do you know if it's claimed?
You'll know that it's claimed because you won't be able to edit it.
So just to clarify what we see, the moment you save it is no one else can edit it. They can see the and only the person who submitted the original revision can continue to edit it. So just so that you can immediately visualize it and make sure you just put a space and let's say it would be submitted for review. You wouldn't have submitted the Google form suggesting this is ready, but you can go and edit and the convenience of you're articulating it and submitting it.
Right, so I guess for anyone on Zoom who hasn't heard what Alex, one of the leaders of this project just said, if you want to claim a variant, you can go in, press edit, submit a small edit, you can claim it as yours and then work on an interpretation. And then once that final interpretation that you submit is finished, you can fill out the Google form. During that time, after it's, you press the initial, submit for an edit, it's claimed for you. Nobody else will be able to go in and try to edit it themselves and claim it for themselves.
Okay, and then we have 2 more questions on Zoom. The first is, generally how time-consuming do you think this will be?
So the amount of time that it will take will depend on you. It'll depend on your comfort with integrating and synthesizing the literature, reading it and understanding it. If you're already a practicing molecular pathologist, I can't imagine it would take more than an hour or two. For students who don't have nearly as much training, it would take longer. That being said, one of the metrics that we are trying to capture over the course of the study, how long it does take you, and if you are improving. My hypothesis is that as you guys move forward in the project, if you are a dedicated contributor, you will get better. And that's one of the things that we're going to try to capture with our bi-weekly surveys. One of the questions will be, on average, how much time did it take you in the last 2 weeks to write one of these interpretations? And so you guys will improve. You'll be able to make use of the Google Sheet, looking at everybody else's comments, all the college comments on everybody else's interpretations, try to use that to craft more high-quality interpretations and avoid the pitfalls that other people have fallen into.
If I may add to Muna's points, there are essentially 3 different points that we'll be helping on. One, on the technical side, there's a user guide that will familiarize you with a platform that says, how can I edit an interpretation? How can I submit it? How can I see what is available? There's already a user guide available. The second point that Muna mentioned is a video tutorial of how you should approach and go through the interpretations. These are not going to be original interpretations that you're going to be adding from scratch. These are already well formed, and if they have revision history, you are already able to see it. But also, when you do submit it, there's going to be a review set of comments, let's say if there are additions that need to be changed. So you'll be able to see not only your review comments from our pathologists, but also additional ones that are available so that you could see a little bit of the approach and the methodology and how to put the updated interpretations together. And hopefully you do expect to, as you gain more experience on this, to deliver interpretations faster and faster, but it's the first time I think it would take you longer than it would take you once you get the hang of it.
Right. So I guess just to reiterate some of those points for the people listening via Zoom, we will be providing a user guide, which will walk you through some of the technicalities of how to traverse the PMKB website. Again, I'll reiterate, I'm going to be posting a video tutorial showing you exactly how to do this, how to use many of the tools that we said are really useful within the workflow of constructing an interpretation. And that'll help you guys have a strong, sort of basis upon which you can begin, developing, skill and comfortability with those tools and with, synthesizing literature. I think one thing that we are working on is getting the library involved here with, basically informational sessions on how to better use tools like PubMed or Google Scholar, and how to abstract as much useful information as efficiently as possible. So stay posted for that. Yes, question?
Is it fair to say that each participant should work on a single interpretation at a time rather than splitting a series of variants and saving for the other to work on them at a specific time instead of piggybacking on them?
Yeah, we would prefer that people worked on variants one at a time and didn't go and claim all the tier 1 variants for themselves and then be like, all right, I'll finish all of these over the course of the next 4 months. Like, no, don't do that. If we find that that's happening, you'll be booted from the competition. There was another question from Zoom.
Just kind of confirming that everyone contributing suggestions would be participants in the study, or kind of that was the question. Confirm if they're participants in the study or more.
Yeah, everyone who contributes an interpretation is considered a participant of the study. I mean, sort of tautologically. You guys are both subjects and participants and potentially study authors, depending on your performance. guys are wearing multiple hats depending on the degree to which you contribute. But we are trying to better understand, again, how does crowdsourcing work in the context of curating one of these large clinical databases that physicians use on a daily basis.
And another question was, is there a word limit for the submissions? How in-depth they need to be?
So ideally, no more than like 6 sentences. These are meant to be short and sweet. Again, this is something that's being put in a sign-out report for, an oncologist. They're going to get back all of the, genetic alterations that a patient's tumor may have, and each of those alterations will have some interpretation. You don't want to inundate them with a textbook chapter on BRAF. You could, but you shouldn't. So short and sweet, and again, we're trying to optimize the creation of one-liners. That would be roughly 2 to 3 sentences long that talk about just prevalence and then prognostic and therapeutic significance. We're really trying to keep it as succinct as possible so that only the important information is conveyed to somebody who has to make quick decisions that are informed by recent literature.
One of the things that you will find is the more time that you spend on interpretation, the more succinct it will be. And in addition to the essentially informational blurb of the interpretation, there is a citation component. So if you search on PubMed, just the PubMed IDs, it would automatically extrapolate the whole citation. The key of the blurb or the succinct point is the citations that you provide in addition to your research supports the evidence of why this interpretation is the 4 points that Muna mentioned: the prevalence, the what was the other ones? I forget.
Prevalence, significance, molecular function, and then how does the variant promote oncogenesis? I think that's a great point. We're not trying to write tomes here. There's a reason why you guys are going to be including all of the references that you use in the process of crafting it. Meant to make a succinct, very high level interpretation of this gene variant, include the references, and those are present for oncologists, molecular pathologists to look into it more if they have additional questions regarding that particular gene variant. Any other questions? Anyone? Nothing from Zoom? Okay. Well, I guess that adjourns this session. Thank you all so much for coming. Looking forward to seeing you all contribute to this study. I'll be reaching out to all of you. I have your email addresses with information on how to register for PMKB, and I'll also reach out over the course of the weekend with the details for the YouTube link for this information session, and also with the video tutorial guide. And I'll periodically communicate any updates as they come along. Feel free to ask me any questions. You all have my email address. Thanks.